Hi, everyone, and welcome back to Teach Me in 10. In this episode, we're discussing one of the most exciting frontiers in modern drug discovery, targeted protein degradation, or TPD. We're joined by Dr. Sam Jensen, Associate Director of Biopharma Strategy, who will discuss how this emerging modality is reshaping how scientists approach challenging and previously undruggable targets by leveraging the cell's own machinery to selectively remove disease-causing proteins. He'll detail how key assays and strategies are enabling degrader discovery and development.
Hello there. My name is Sam Jensen. Today I'm going to teach you a little bit about targeted protein degradation and specifically some of the workflows and assays that are used during the TPD drug workflow. So to start off, what is targeted protein degradation as an intro to this space? So this is an emerging drug modality that involves a small molecule. What's interesting about targeted protein degradation is that it enables a really rapid and specific degradation of a target protein of interest. And it does this by harnessing the internal cellular machinery, specifically the ubiquitin proteasomal degradation machinery. Now, this machinery is typically utilized for degradation of various proteins during a normal cell's ongoing function. In the case of TPD, it's harnessed to deliver a targeted degradation. And there are a variety of TPD platforms. So you have this thing called in red, which is either your PROTAC, it also can be called a degrader. Again, this could be a molecular glue. Essentially, this is a small molecule that has two different binding domains. So on your left side, this is the binding domain that is going to target your protein of interest, so the thing that you want to degrade. All it needs to do is be able to bind to that protein and bring it into coordination with the other ligand binding domain, which is to bind on the right side to an E3 ubiquitin ligase. So essentially, again, it has two binding domains, one to the protein target of interest that you're trying to degrade, and one for the E3 ubiquitin ligase. Typically used are E3 ligases such as cereblon or VHL. So to visually see what this looks like, when the PROTAC binds the target protein in blue and the E3 ligase together, what happens is it brings them into proximity with one another. And that proximity allows for the E3 ubiquitin ligase to carry out its normal degradation function, which is to add ubiquitins to the target protein of interest. Now, when it adds those ubiquitins to the target protein of interest, it is then signaled for degradation by the proteasome. So again, this allows for rapid and specific degradation of a target protein of interest, and your PROTAC or degrader is then recycled back and can degrade additional molecules. So with each step in the workflow, there's different assays that are employed. And so what I'm going to do today is walk through those various steps and talk a little bit about some of the assays that are typically used to assess the functionality of your degrader at each step in the process. So starting with the first step, we really have this degrader discovery. Now, there's kind of two parts of this degrader discovery. It's actually, developing a degrader, discovering a degrader that's going to be able to engage your targeted protein of interest as well as the E3 ligase. The other one really hinges on the fact that you need to understand the levels of E3 ubiquitin ligase in your cells or tissue. This is important because you have to have a certain amount of either cereblon, VHL, or another E3 ubiquitin ligase in the cell in order to harness that internal machinery to then degrade the protein of interest. Another thing that people oftentimes do is to discover accessible nearby reactive residues on the targeted protein of interest. Now, this can help with the degrader design and improve the degrader binding strength. Antibodies can be used that are highly specific against either cereblon, VHL, or other E3 ubiquitin ligases. This can be done in cells, in tissue, in multiple applications. And then the reactive cysteine mapping and profiling can be done using mass spectrometry. For the second step, what you're looking for is something called ternary complex formation. So essentially, this is where your degrader is taking and binding to your protein of interest and bringing it into coordination with the E3 ligase. Now, this is a very critical step because the the formation of this complex is going to lead to the ubiquitination and ultimate degradation of the protein. So what the most commonly, way that someone would want to confirm that this ternary complex is indeed taking place is through FRET. So TR-FRET essentially utilizes europium cryptate, and specifically it's usually utilized on europium cryptate labeled antibodies. This can enable biochemical assays where you recombinantly express, say, your targeted protein of interest with a flag tag, recombinantly express your E3 ligase with an HA tag. Then you can look at those things, mix them together, and try a number of different degrader designs to see which ones form the best ternary complex. And then cell-based assays can also be used where you're actually using an antibody labeled against the protein of interest that targets that, as well as the E3 ubiquitin ligase, for example, cereblon. That way you can see in an endogenous format how these things work together to get an understanding of how well that ternary complex formation occurs. After you've determined ternary complex formation, you then need to see whether or not the target actually gets ubiquitinated. So how do you do that? So again, what you're trying to do here is really to both identify that your target of protein was ubiquitinated and also quantify that. So there's a couple different ways one can do that. So one of the most common ways this is done is through ubiquitination sites via LCMS. So essentially here what you can do is you can enrich and pull down with an antibody against a ubiquitin site, pull down all the proteins that are ubiquitinated, then read them out by mass spec to provide both their identity and quantification. Another thing that is used are these tandem ubiquitin binding elements, or tubes. So tubes are essentially a protein that has multiple ubiquitin binding sites. And what it allows you to do is really to profile pan-ubiquitination or branched ubiquitination. And then finally, there's individual antibodies that can detect either singular ubiquitin or various sites of a ubiquitin branch. Taken together, these allow you methods to both, again, identify whether or not your protein is being ubiquitinated, but also to quantify the amount of ubiquitination. The next step in the process is after it's been ubiquitinated, you obviously want to see, is my protein of interest actually degraded? So in this step 4, what you're really going to do is look for the loss of your protein. So that is typically done, over time, over dosing amounts, a number of different parameters, and it can be read out in a number of different applications. This is typically done with Western blotting because you are looking for the degradation of your protein of interest. So Western blotting, simple Western by Protein Simple is very frequently used to assess degradation, but it's also done in things like high content imaging and analysis. You can use ELISA and other types of pair-based assays to quantify that level of degradation, as well as flow cytometry. To determine next, after you've shown that you're able to degrade the protein, what are some of the on and off-target effects? And when we're thinking about off-target effects, we're really trying to think globally across the proteasome. We want to make sure that we're understanding any things that might be happening that are not related to the degradation of our target of interest. So one of the most common ways that people look at this is global profiling, global proteome profiling by LCMS. So essentially, before drug, after drug, what proteins go up and down? Are other things changing in response to that degrader? And if you want to look more at specific post-translational modification sites, so whether it's a ubiquitination site or whether it's a phosphorylation site related to signaling and many more things, then you can use antibodies to pull down those specific post-translational modifications and assess them by LCMS. And finally, you also want to understand the phenotypic response of the cell to the degraders. And so to do that, what you're really going to be doing is assessing downstream readouts that fall downstream of that target to ensure the functionality, and also phenotypic changes within the cell following degrader treatment. This is where you're really looking at a wide plethora of different types of antibodies that are going to basically measure, depending on the biology of what you're trying to target, various readouts and phenotypic markers to look for, say, increased or decreased signaling within the cell, changes in the cell morphology, changes within the cellular state and identification, things like that that tell you that you're getting the predicted phenotypic response that you would expect. And then there's obviously your functional cellular assays. So these would be things like, a cell death assay, cell viability assays, cell cycle assays, a number of different cellular functional assays that determine the phenotypic change following degrader treatment. Let's look to the future for a second. So what's next for TPD? So the first thing is many more TPD drugs will continue to enter clinical trials. Some of those will ultimately win FDA approval, and this will broaden the reach to patients. There's also a very quickly emerging field of non-degrader applications. This is also known as induced proximity. So if you can bring two proteins together, but not necessarily for degradation, that opens a number of doors to potential possibilities that include things like the specific dephosphorylation at specific sites of proteins, for example, bringing transcription factors into the nucleus, and many more possibilities. With the advances of artificial intelligence, AI-driven design will also aid this field. So that's not just in terms of in silico design of the degraders, but also as well as the target ID. And essentially, this is going to streamline and reduce some of the necessary preclinical testing and money spent in developing drugs. And then finally, there's an expanding number of applications. So since TPD is really agnostic to the disease or indication type, it can really be employed for additional indications where any type of protein degradation is needed. So there's a lot in the future and a lot on the horizon. It's a very exciting space to learn about, and I really thank you for your time in listening to this Teach Me in 10. Thank you.
Thank you for joining us for this episode of Teach Me in 10, brought to you by Cell Signaling Technology, who are a different kind of life science company, founded, owned, and run by active research scientists. CST offers a comprehensive suite of platform-compatible, application-validated tools that help answer critical questions at every stage of TPD drug development. And with in-house manufacturing, you can rely on consistent quality and reliable supply. If you like this episode, don't forget to like and subscribe to catch all our latest releases.